So chapter 4 is named The Extensions and Modifications of Basic Principles. We're talking about Mendel's principles here. So remember, Mendel came up with ideas of dominance and recessiveness, and he looked at genes, although he didn't realize it, that were not linked. And so Mendel lays out a lot of basics, but now we can now look at some extensions, some added knowledge that we have since the time of Mendel. Though half of this chapter is going to deal with things relating to sex and sex chromosomes, gender. So we start off with this statement: the rules for sex determination are not universal, meaning they do not only, they're not the same for all organisms. Mammals have different rules than many other groups of organisms. So just because humans and mammals have an XY chromosomal system of sex determination, of determining what makes, what is a male and what is a female, other organisms have different systems. So ZZZW is a system that is chromosomal, found in birds, and in that case ZZ, having two of the same type of sex chromosomes, is the male, and ZW, having one of each different type, is the female. And so they use different letters to help that you recognize that. And then XXXO is something we saw in lab with the grasshoppers, because the female grasshopper had 2 X chromosomes in each cell and the male only had 1. And so those are chromosomal systems. But there are insects that use a haplodiplo system, where the males grow from an unfertilized egg and they're completely haploid, and a female will only grow from a fertilized egg. So that's a haplodiplo or haploid-diploid system. We are going to look at the XA ratio system found in Drosophila, since it's one of your model organisms in this course. And then reptiles, actually reptiles are all over the map. Some of them use chromosomal systems, but some of them use a temperature-dependent system in which it depends on what temperature the egg is incubated at, what the final sex of the individual is. And so you have all these different, and there are more than this, but these are, some examples of different ways that sex as a phenotype is determined even from the early embryonic stages. We will focus, though, on the X and the Y chromosome, since we are interested in humans. The X and the Y chromosome are really not homologous. They don't have the same genes on them. Most homologous chromosomes, as a rule, have the same genes in the same order, even if it's not the same alleles. But the X and the Y don't. In fact, the only thing similar about X and Y are found at the tips, called the pseudo-autosomal regions, and those are the parts that will actually synapse together during meiosis to allow this to become a tetrad. So in humans, and like I said, what's true in humans is true of mammals, but not of the broader group of organisms, just mammals. In humans, the presence of the Y chromosome is the trigger for male development. So you can think of the Y chromosome as being the trigger. If you don't have a Y chromosome, the default path is female development. So you need to memorize that rule. And it's not the entire Y chromosome, it's actually just a certain section of the Y. It's called the sex-determining region of Y. And this area here is actually where the gene is that is the trigger. So if you don't even have to have a whole Y chromosome, you can just have, there are individuals that just have a section of the Y, and it's enough. As long as it contains this SRY gene, then it will trigger the male development. Now, some evidence that this rule is correct is found in individuals who have aneuploidies, which means they have too many or too few chromosomes compared to what is considered normal. And this is an example called Klinefelter syndrome. It's a male with 47 chromosomes per cell, and the sex chromosome makeup is XXY. So the extra chromosome is typically we consider there to be an extra X. And so it's a male with an extra X chromosome, but you can see that having an extra X still phenotypically gives you a male. There are sometimes some fertility problems for a Klinefelter syndrome male, but until that point, you could have a person who doesn't even know they have this chromosomal aneuploidy until they're trying to conceive a child. Now, the flip side of the coin is called Turner syndrome. It's a female who has one X and then that's it. So each cell only has 45 chromosomes, but one X, it turns out, is enough to create a phenotypically normal female, except there is a little bit of a giveaway on this case. In Turner syndrome, you will often have what's called the webbed neck, W-E-B-B-E-D, webbed, and that just means this extra tissue that you can see. And I picked a picture of a female it's pretty pronounced. I have seen individuals with Turners that have a less pronounced webbed neck, and if you wear hair long, you know it can be not very noticeable. But this one is very noticeable, and the webbed neck is a clear signal for a Turner syndrome situation. So when does sexual differentiation, when does an embryo start to become male or female? It's hard to say exactly, but by the 5th week of gestation for a human embryo, you can start to see 2 clumps of tissue we call the gonadal primordia, meaning they're the primitive sex organs, so they're either going to turn into 2 ovaries or 2 testes. And if the cells contain a Y chromosome, they'll turn into the testes, and if they don't, they'll turn into ovaries. Like we said, the female is the default, so only if there's a functional SRY gene will you get the male. Now, the functional SRY gene is just the trigger. It triggers several other events, which trigger, in turn, other events, which eventually result in male development. But you've got to remember that male development includes a whole bunch of neurological things and genitalia type things. And so to actually define what is a male and what is a female actually gets pretty complex. But notice that I've underlined the word functional, because you could have an individual that has an X and a Y in each cell, but the Y chromosome doesn't have a functional SRY gene. It might have a non-functional or mutant SRY gene, in which case the individual would develop it completely as a female, because female is the default. So the SRY gene has to be present and functional to have this cascade of events happen. The problem is, even if this cascade of events is triggered by a functional SRY gene, if any of these next steps is non-functional, mutant, then you can still have a problem with part of the male phenotype. A problem meaning a situation where the brain development doesn't really match the genitalia development or something along that line. And so this is what I call a malfunction or disorder of sexual development. I'm not going to go into a lot of detail here. You can do some research online, but the point I'm making when you read through these slides is there are documented, pretty well studied scenarios or situations where somebody has a mutation of a gene, and it's going to be one of the genes that the SRY gene will activate. And when those genes are mutant, you end up with a partial failure of male or female development. So congenital adrenal hyperplasia is an example. You may get an ambiguous external genitalia. And of course, when you have a baby, even though the brain might be androgenized, meaning it's going through a male development, a baby can't tell you that. So sometimes decisions are made with a baby, and later on the individual feels like their body, their genitalia is a mismatch for what their brain is feeling like. This is complete androgen insensitivity. You have somebody who looks completely like a female, it completely defaulted to a female, even though the cells can be, you can look at the chromosomes and see that there's clearly an X and a Y. And so one explanation for that is the androgen receptors are mutant, they're not functional. Then you can get something like a partial androgen insensitivity where the individual is XY, but the phenotype is not clearly male or female. And again, sometimes in those cases you have some ambiguous genitalia. Now, if you want to know what ambiguous genitalia is, you can Google it. Google image gives you some pictures. And so babies come out sometimes that don't have a clear, a penis or a vagina. They're sort of in between. And so that does happen, and these things are biochemical in nature. It's a failure not of the SRY gene, but of one of the later events. Now, in Drosophila, sex determination is based on a ratio of number of X chromosomes to number of autosome sets. So if you're haploid, that's one set of autosome. If you're diploid, that's two sets, etc. And so if you have two X's and a diploid cell, then that's 2 to 2. That's a 1 ratio. 2 to 2, 2 over 2 equals 1. If you have an individual that has an X and a Y and two sets of autosomes, that's a male, but not because of the Y, but rather because you have one X over two sets of autosomes. That's a 1 over 2. That's a ratio of a half. So these are the rules here. I'm just showing you what the rules are. And the way we know the rules are correct is that when you can have a fly that has one X and that's it, and two sets of autosomes, which is a 1 over 2, and that does give you a male. So no Y chromosome, but still a male. So it tells you that it's not running, the flies don't use the human rules. Flies have fly rules. This one is XXY and two sets of autosomes. In humans, this would be a Klinefelter's male. In flies, this is a perfectly normal female. So this is, a different rule. In Drosophila, it's about X to A ratio. And instead of ratio, I'd make a fraction out of it. I think that makes it easier. Because in the end, it's really about a fraction. The number of X's on the numerator, the number of autosome sets on the denominator.